I'm Pam Feliciano. I'm the scientific director for Spark, and I'm really pleased to be here with you today, and thank you for taking the time. And we're really excited to have the opportunity to explain Spark and the kind of data that we're able to share with our participants. So in today's talk, we're going to we are going to review what Spark is, and I will go over the process for that we have in place to return genetic results to our participants, and also give an update on where we are with our genetic research. And then I'll turn it over to Kiely, who will talk about the other kinds of data that we're returning to our participants. Okay, great. So like most of you today, most of you are here because you have a personal connection to autism, and so do I. My son was diagnosed 11 years ago, and it really changed the trajectory of my life and made me want to work in autism. So 5 years ago, in September 2013, I joined the Simons Foundation and helped launch Spark in 2016. And what drives me is what drives a lot of you. I'm interested in understanding autism better. I want to make a difference and help families, and that's who I am. Okay. So just in case you're not exactly sure what Spark is an effort to build a really, really large research community of individuals with autism plus their family members, and connect them to researchers so that we can speed up the understanding of autism. And what we're doing is we're collecting DNA, that's genetic material, from families, as well as medical and behavioral information from those people, and we're sharing that data with qualified researchers and connecting you guys to those researchers for other autism research studies. And our goal is to lead to better understanding of autism and discover things that haven't been possible before, and we want to share everything that we find back to you. So that's our goal. We also have a slide here that explains what Spark isn't. I think that there are a lot of misconceptions about what research is, and we wanted to be just real clear about what we are not. And so we want to be clear that we are not looking for a cure for ASD. We are interested in helping develop better treatments to support people with autism better and to improve their lives and the lives of their families. With regard to genetics, we are not a genetic testing service. We are only able to return genetic results to 10 to 20% of participants, and that's not because we know more. This is just basically the limit of what we know. And to date, we've only been able to find genetic findings in 10% of our participants. This will increase over time, especially as we really are starting to grow now. But right now, we are only able to return or identify genetic findings in 10% of our participants. And so we also want to be clear about that. And we are not developing genetic tests for autism. We are analyzing everyone's genetic data using state-of-the-art methods. We're discovering new things, and we're returning those results to families as we have enough data to do so. But we are not in the business of developing any kind of tests that could be used by families to do other things. So we're just analyzing the data that we get from our participants and returning it to them and discovering new things and sharing that data with our participants and with researchers. Okay, so anyone who has a professional diagnosis with autism is eligible to participate in Spark. All you have to do is go to our website at sparkforautism.org, and that's where you can enroll in Spark. And you have to live in the United States, be able to speak English, and you can invite your first-degree family members. So both of your parents and all of your biological siblings can also enroll in Spark. We also wanted to have a few statements about how we protect your data. Data security, especially in this day and age, is becoming a very big concern and a topic of real importance in the news. So we wanted to be clear about what we do with your data. Before we share data with researchers, everything that we have from you is stripped of personally identifying information. So things like your first name, your last name, your address, your zip code, your date of birth, all of those things are taken out from the data set that we share with researchers. That information is held securely in multiple physical servers that in locked facilities here at Spark and various places around the country. But anything we share with researchers will be stripped of any personally identifying information. Researchers can also only access your data if they sign a legal agreement that they will protect your data and only use it for scientifically valid purposes. We also, in addition, hold something called a Certificate of Confidentiality from the federal government agency called the National Institutes of Health. And that further protects your data because people who are not researchers, such as people in the criminal justice system or a non-qualified researcher, cannot access your data without your consent. And we also want to just be clear that when we inform you of other research studies, we tell you about them, but we will not send your information to those researchers that want to recruit you unless you say you want to participate in that study. So we just wanted to be very transparent about what we do with your data and what isn't possible with it. Okay. So in terms of what participation looks like, as I said earlier, you just go to our website at sparkforautism.org. You choose whether you want to participate in the genetic analysis. You can also opt to just participate in the data portion, which would just mean filling out questionnaires. And you also, no matter what option you choose, you will always be able to be contacted about new research opportunities. And again, no further research is required. We will just send you an email and say, would you like to participate in this study? And then you say yes or no. And then you can also hear about things like this webinar and other events that we have to try to give back information to participants. Okay, so in terms of where we are with recruitment, we've grown a great deal in the past 32 months. We've recruited 53,000 individuals with autism into Spark, which is really, really exciting. And I think around 100,000 of their parents and siblings have also joined Spark. We've collected saliva from 80,000 people. So that's 80,000 milliliters of saliva, which is a lot of spit, which is really exciting. And together, all of you have filled out 210,000 questionnaires. So we have many, many, many millions of data points. So this is very exciting. And what we want to do with all of this data is be able to return that information to you, including the genetic results, and also the phenotypic results, which we'll talk about today, so that researchers can contact you based on either your genetic results or perhaps some of your phenotypic traits and match you to research studies. And this way, we're going to be able to accelerate better research because we all know that autism is extremely heterogeneous, and research would benefit by being able to study more homogeneous groups. So this is what our mission is, and we'll be talking about the efforts and the inroads that we've made towards this bigger goal. Okay, so genetics. I'm going to have a few slides just explaining autism genetics. Autism is actually very heritable. We know that autism is more genetic, has more genetic factors or more genetic influence on it than many, many, many other common diseases. And we know this by looking at twin studies. So if you look at hundreds of twin sets that have autism, and if you only look at twins that are monozygotic or also, called identical, if one has autism, the other twin, 77% of the time, also has autism. If you look at fraternal twins, which do not have the exact same genome, they have the same amount of genetic similarity as siblings. Their concordance rate, or the rate of finding both with autism, is 31%. So if you have one twin with autism, the other one has autism 30% of the time. And if you look at siblings, that rate is 20%. And what this data tells us is that because the 77% in identical twins is higher than fraternal twins and siblings, that tells us that autism has a very large genetic component. And then if you compare the fraternal twins to siblings, these individuals have the same amount of genetic similarity, but the rate of autism concordance is higher in fraternal twins because they share the same prenatal environment. So that tells us that autism has both a genetic and an environmental component. And for the past 20 years, researchers have been studying autism and trying to identify genetic factors that cause autism, and we've made a lot of progress in the past 10 years, and so we'll talk about that. Okay, so how much of autism, how come I didn't? So autism, how much of autism is genetic? It's not moving. Oh, okay. Very weird. Okay, so how much of autism is genetic? So people have done a lot of studies, not just twin studies. Most recently, there was a really, really large study of every child in Sweden born in a certain time period. And the basic conclusion from this study is that perhaps up to 80% of autism can be explained by genetic factors. And the rest would be explained by environmental factors. There are other studies that conflict with that percentage, but I think it's fair to say that autism, at least half of autism could be explained by genetic factors, with the rest being explained by either environmental factors or a combination of both genetic and environment. And so for the past 10 years, people that Safari has funded and ourselves have been studying what exactly are those genetic factors that lead to autism. And so on the next slide, what we have is a summary of what we've learned. So in the 1970s, the twin study showed that autism is very highly heritable. And there was some progress. So early 90s, there was a gene discovered for Fragile X called FMRP. A few years after that, the gene for Rett syndrome was discovered. That's MECP2. And then 10 years after that, more work was done, and the amount of autism we could explain 7 to 10 years ago was probably about 5%. So there were genes, also this small number of genes discovered. We can talk. And then today, in 2018, the total number of autism genes that we know about is roughly 100. And altogether, these explain between 10 and 20% of autism. There are many different ways that genes can lead to autism. And so we'll go over the different kinds of heritability patterns known to exist in autism. So many of you may not have heard about changes that are called de novo. This is when a genetic change happens in the egg or the sperm that produces the child. So here we're calling de novo means new, and that's what this is referring to, because the genetic change is only present in the child, and even though it's inherited from the egg or sperm from the mom, it's not transmitted in the inherited in the traditional sense, because it's not like the mom or the dad has that genetic change in their body or in their brain. It's just in their egg or sperm cell. We've found, surprisingly, in the past 10 years that these types of genetic changes explain a lot of autism. And you can see, though, in order to determine if something is de novo, we need not just the child, but also both parents. This is why there's such an emphasis in Spark in recruiting complete families, so that we can do the best genetic analysis we can and have the highest opportunity to return a result to a family. In other cases, genetic changes are so-called X-linked, This is when a gene variant exists on the X chromosome. In most of these cases, the transmission is occurring from the mother to the affected child. So you'll see here on the top here, the XY would be the unaffected father. The XX would refer to the mother. And you can see in the diagram below, a child who would be affected would most likely be a male. So he has the X chromosome with the change from the mom, and his other sex chromosome is the Y from the father. And because he only has one copy of the X chromosome, which carries the variant, that child would have autism. And we already have seen cases of this in Spark, and we've already started to return results like these to our families. Sometimes genetic changes are inherited, and that's when a genetic change is coming from either an unaffected parent or sometimes a mildly affected parent. And so the variant can come from the mom or the dad, and the mom or dad doesn't necessarily have to have autism. And the child inherits that change but has autism. And this happens when we have genetic factors that aren't fully 100% penetrant. And that means that the genetic changes can be carried by a parent and transmitted to siblings, and sometimes they cause autism and sometimes they don't. This is trickier. We need a lot more data to be able to identify new genes that are like this, but we are starting to do that, and it's really exciting, and I think this will contribute more and more to the results that we're returning. I wanted to try to explain exactly how we identify genes that cause autism. It is a complicated process, but I'm going to try to explain it in a way that's relatively simple to understand. Okay. All right, so the animations didn't transfer, but that's okay. So the yellow circle, if you just sequence everybody, on the webinar today, there probably are people with autism, but if you sequenced everyone who doesn't have autism, you would find thousands of genetic variants in every person. And when you sequence people with autism, you also find variants, and most of them overlap. So most of them have no significance for autism. They control other things. They can control nothing. They could just be, changes that are present. They could affect hair color. They could affect height. They could affect eye color. But when you sequence enough people with autism, the stuff that is not overlapping with healthy people, but repeatedly found only in people with autism, are the things that likely cause autism. Okay, so I don't want to say never. They're never found in people without autism. I guess there are different degrees of neverness. But what I can say is that if we have enough people with autism, we can start to identify genes that are recurrently found in people with autism, but not found in people without autism. And that's basically how we're able to figure out what genes lead to autism. The bottom square, where I'm talking about variants that are found repeatedly in people with autism, but rarely in people without autism, those would be the variants, okay, I don't have a pointer. They would basically be the variants on the edge of the yellow circle and blue. So they're rarely found in the yellow circle, found in the blue. So maybe out of 50,000 people with autism, you see that variant 100 times, and the same number of people you would see it 3 times. And we can make conclusions from that statistical difference and say that that variant must affect someone's autism risk because we find it way more in people with autism versus people without it. And those are the kinds of things, variants that are inherited. The ones on the top where I'm talking about are almost never seen in people without autism. Those are things that are much more penetrant, and a lot of times they're de novo. So this is the process that we are analyzing everyone in Spark. And I wanted to sort of explain the complexity of this process because it's never super black and white. And the more data we have, the better conclusions we can make. And So this is what we're doing to identify variants in genes that are known in autism and also to discover new genes that lead to autism. I also wanted to sort of give an example give a summary of where we are. So on the top, what you see is that we've completed analysis of 457 families. You're going to hear me say the words trio, quad, and duo, and when we're talking about a trio, we're talking about an offspring with autism plus their parents. And out of these 457 families, 10.5% of them have a returnable result. We've returned about half of them so far, and the other half we will be doing throughout 2018. We've also identified another additional 4% that have potential results. So these are ones that we're not exactly sure whether they're found repeatedly enough in people with autism and not without. And so as we have more data, we'll be able to make better conclusions and decide which of those 3.5% actually have genes that we can confidently say lead to autism risk. So that will take some time. So you can see as we have more data, the more information we can return to families and the more information we'll be confident about. So in total, 48 families will have received or will receive results in 2018. We currently have 7,500 families that are in analysis. So at Spark, what we're doing is we're accumulating many thousands of samples and sequencing them in large batches. And this is for many reasons. It's a lot of logistics to coordinate the transfer of tens of thousands of samples to the sequencing facility. We also get the best prices when we have 30,000 samples to sequence, and that enables us to sequence the most families. So in the past year, our sequencing facility has sequenced 7,500 families. They have returned that data to us, and we are in the process of analyzing that. Most of those families are trios, quads, or duos. There are a few that are very large families, but those are the minority. And based on what we've seen in our first analysis, we expect to return results to about 750 families next year. We're starting that analysis, and we're in the process of just confirming all of the results. We also have accumulated DNA from 6,500 families, and those are in progress. So the DNA has been extracted, and they are going to be transmitted to our sequencing facility before the end of the year, and we'll get that data back next year, and I would expect to be able to return results to additional 650 families at minimum, beginning at the end of the year through 2020, and this is all of the sufficient DNA that we've accumulated as of 2 months ago, and the cycle is continually being updated based on what we're getting back from you. I wanted to give a little more information about the kinds of results that we've returned. You can see that 70% of what we're returning are de novo changes. So these are changes that we're only sure about because we have the child's DNA and the parent's DNA. 1/3 of the results are inherited. I want to say that the percentage of the results that we're going to return does sort of depend on what family members we have. The more family members we have, the better chance that we'll be able to discover something, but not necessarily always. So we will be able to return some information to the parent-child duos and the individuals who don't have family members participating in Spark. I would just say that the percentage of those families will likely be less. And it's really hard for me to say what that will be. I think we need to analyze these families and see where we're at. I will say, as this is an iterative process, as we collect more and more data, we're going to be able to return information to families. But for the next year or two, the people that we're able to return, most likely to be able to return information are the families with both parents and even more when there's another child, whether they have autism or not, in the study. Okay, so So I just wanted to make the point that discovering new autism genes is most effective if we have DNA from parents and siblings. And we're really looking forward to analyzing all of the data that you've been kind enough to share with us, and we hope to start returning hundreds of results in the next few months. Okay, and so we have one video. We won't play it now, but if you haven't seen it, this video, the link to it is right at the bottom of the screen. It explains the return of genetic results story that one family got through Spark. It's a really great video, and I encourage you to watch it. So that's my part, and I'm happy to transfer over to Kiely right now.
Okay, thank you, Pam. Irene, if you can go to the next slide. Hi, everyone. My name is Kiely. Like Pam and also many of you listening in, I also have a personal connection with autism. My son Isaac was diagnosed with autism back in 1996, so about 10 years before Pam's son. At that time, there was less known about autism, and also there was very little research being done, and that was extremely frustrating as a parent. So eventually, I was able to translate that frustration into motivation, along with many other parents, as well as researchers and clinicians. And in 2006, I was able to help start the Interactive Autism Network, known as IAN. And so now, 12 years later, we are merging our efforts with Spark so that we can work together towards this common goal of advancing autism research. Next. What I'm going to talk about is other ways that Spark is sharing results with families. And so, one of the common criticisms that I've heard from families who participate in research is that I participated in that study and I never heard anything back about what researchers learned. So Spark, in many ways, as Pam described, is committed to changing that paradigm. We know that participants spend their valuable time sharing with us important information about their autism journey. And so also, as Pam mentioned, it's going to take a while before we get through all of the genetic analysis. And, we're not going to be able to return genetic results to everyone. But there are things that we can return to everyone, and those are, I'm going to talk to you about those today. And first, I'm going to talk about our data snapshots. We've published 2 today. The first one focused on our first 18,000 families or participants with autism who joined Spark. And then the second one focused on the independent adults with autism in Spark. Okay, so the first snapshot, it focused on the first 18,000 participants with autism. And for those of you who may not be familiar with research, this is a huge sample size. Having lots of people participating in Spark allows researchers to investigate many different topics in many different subgroups of individuals. And although the number of participants in Spark has now tripled, the findings in this report are actually still relevant and very important. This first report also gave us a look at how Spark participants are similar and how they may be different from what we already know about autism. So next. So here's one of those. So here you can see the enrollment in Spark by sex. So we've known for many decades that boys are more likely to be diagnosed with autism than girls. And the ratio that is reported is 4 to 1. And that means that for every 4 boys diagnosed with autism, only 1 girl is diagnosed. And so as you see on the left, the male to female ratio of children in Spark matches that of the overall autism population. This gives us a clue that Spark is doing a good job at recruiting a representative sample of individuals with autism. Now, if we look on the right, in adults with autism, we see a male to female ratio of 2 to 1. And so there are many factors to consider when exploring why we see this difference. So first of all, just in this very simple analysis, we looked at all of the adults in Spark who have autism. And that includes individuals who are dependent, so they had a parent or guardian enroll them in Spark, as well as individuals who are independent. And so if we actually break down those two groups, what we find is that in the dependent adult group, we see a ratio of 4 to 1, similar to the children. But in our independent adult group, we actually have a 1-to-1 ratio, which means we actually have proportionately more females with autism choosing to enroll in Spark. So this is really great because it gives us an opportunity to explore autism in females, which has often been understudied in research. But it also means that we probably need to do a better job at recruiting more males with autism who are independent. Next. Okay, so this slide shows the average age of autism diagnosis for both children and adults in Spark. So if we look at the kids first, we see that on average the boys received their diagnosis at 4.1 years of age, and the girls were slightly older at 4.4 years. And then if we look at the adults, we see a much larger age difference at the time of diagnosis between males and females. And although we would need to do a more rigorous analysis to explore this difference, what we do know is that our results are in line with what has already been reported in the literature. Others have found that girls are diagnosed later than boys. This could be because autism symptoms look different in girls than they do in boys, and perhaps the diagnostic tests and clinical training used to diagnose autism isn't adequately identifying the disorder in girls. Next. The language development varies considerably in individuals with autism. Some individuals are fully verbal and able to communicate in complex sentences, while others are mostly nonverbal or use single words to communicate. Again, other studies have reported that approximately 25% of the autism population is minimally verbal. And so if we look at our numbers here and we combine the 13% who do not speak and the 12% who are able to use single words, we get a very similar number. This type of data is very important to autism researchers. If you think to some of the genes that Pam talked about, there are autism genes that are associated with significant language impairment, and then there are other autism genes that language impairment does not happen. And so researchers are able to use this type of information along with the genetic information to learn more about how the brain functions and which pathways and proteins are involved in language development. Spark participants also report on other medical conditions and symptoms that they have. So for some individuals with autism, it's actually these co-occurring conditions that are more challenging than their core symptoms of autism. And it's also very important that clinicians who provide care to individuals on the spectrum look at these other conditions and address them as well. So if we look at the children on the left, we can see that depression is quite low at 5%, and then anxiety a little bit higher at 16.5%. But now if we look at the adults with autism, we see that roughly a third have depression and the same for anxiety. However, if we look at something like ADHD, it's actually similar in both groups, 32% in the kids and 35% in the adults. And so there's actually a lot of research now looking at the overlap in autism and these other psychiatric conditions, and we're learning that some of the same genes involved in autism are also involved in ADHD, mood disorders, and anxiety disorders. Next. So again, this first snapshot, there's lots of different topics covered. If you haven't seen it, I would encourage you to go to our website and take a look. Next. So now I'm going to spend a little bit of time talking about the second Spark snapshot, which was just published 2 months ago. It focused entirely on the independent adults with autism in Spark. At that time, we had close to 2,000 individuals. And historically, research has not really focused much on the adult population, but this is changing. And as I move through the next few slides, I also just want to point out that within this group, about half of the adults, independent adults, were diagnosed in childhood, and the other half were diagnosed in adulthood. So next. So as I mentioned earlier, here you will see the breakdown of males and females with autism in this group who are participating in Spark. And we actually have a few more women than we do men. You know, this may be because women are actually, even in other diseases, are more interested in participating in online health research. And so that may be one of the reasons that we have more enrolled. Next. So here's the age breakdown of independent adults in Spark. So you can see that a lot of our participants are in their 20s and 30s, but we also have quite a few people who are 50 and older. Education level varies a great deal. However, if we look at the last two circles, 38% of the independent adults in Spark have a 4-year college degree or higher. So in general, higher education leads to increased employment and better job opportunities. But we know from other research in autism that adults with autism are actually more likely to be unemployed or underemployed. So this is also important data as we explore why that is and try to develop supports for individuals on the spectrum as they move into employment. Next. In this report, we also look at sexual orientation and gender identity. So you'll see here that 60% of the adults registered identify as heterosexual. So sexual orientation and gender identity are both topics of growing interest in the medical research community. Both of individuals who belong to sexual and gender minority groups are underrepresented in medical research, and we've known from the few studies that have been done that they are at increased risk of poorer health outcomes. And so this is an important topic that as we develop effective services and think about the entire person with autism, that these are other things that we need to be considerate of. Next. And so again, there's a lot of other topics. I can't go into all of them, but I hope you'll take a look at the snapshot, which you can find on our website. Okay, so now I'm going to switch gears a little bit, and Pam mentioned Research Match. So that's the program within Spark where we let Spark participants know about other research opportunities. It's pretty simple. We always reach out to you first by email to let you know about the study. Step number 2, as Pam mentioned, you decide whether or not you want to participate or not. If you do, your participation will further advance research and lead to new findings. And then to close that loop, we work with the researchers to develop reports that are family-friendly that we can send back to you and also share with the broader autism community. Next. There are many different types of studies that are working with Spark. They cover many different research topics. There's pretty much something for everyone. I do want to say that all of the studies undergo a review and approval process to make sure that they are ethical and that they have scientific value. And because there are so many different studies, we are careful and mindful about how often we contact people. We want to make sure that you have the opportunity to participate, but we also want to be respectful of the burden that research can have sometimes, and we want to make sure that we don't fill up your inbox as well. Next. So here's an example of one of the first Research Match studies that Spark helped with, and this is from the preliminary report that we prepared with the researchers. This study actually recruited families last summer. The researchers are based at Johns Hopkins School of Public Health, and they're really looking at that interaction, that combined genetic environment interaction, to better understand what is happening in that little part of the overlapping circles. This part of the study was actually completely online. Mothers were able to complete a 20-minute survey that asked about their health before, during, and just after pregnancy. Next. So after the initial data was collected, we worked with the researchers to develop a preliminary report. So in this report, we provided more background information about the study. We also presented a few summary stats, and we provided information about how the researchers would be using the data to explore the questions about gene-environment interaction. 1,200 families participated in this study. That was a tremendous response. Over 60% of those who were invited participated. As part of the study, the researchers also collected information about geographic location during pregnancy, and so the study team will be able to use this to explore environmental factors outside the womb that may be associated with autism. The next, also in the report, we provided just a couple of summary stats from the pregnancy information. The researchers are actually going to be doing a much more rigorous analysis of all of the great data that people provided. And then when they do their final publication, we will also be reaching out to participants to let them know what was discovered. Next. Oh, so every study, every Spark Research Match study that we do has a very short 3-question survey at the end for participants. Your feedback is very important to us. And we ask you to rate this topic both on the importance of the topic to you as well as your experience participating in the study. And then we also ask families, they can provide any additional comments about their experience. This particular study got very high ratings on both questions. Next. So to date, we have 37 studies that have been approved and that are partnering with Spark and you to further accelerate research. And over 35,000 Spark participants have been invited to at least one of these studies. And so I have just a couple more slides. So all of this great stuff you can find on the sparkforautism.org website. So I would encourage you to go there and look under the Discover button. And you can find research news and articles. You can learn more about autism genes. We have interviews with researchers as well as inspiring family stories. You can find links to all of our webinars. And we would encourage you to sign up for our newsletter. And also, we'd encourage you to follow us on Facebook and Twitter. Actually, Vincent posts new posts every day on Facebook, and that's actually a great way to stay up to date on what's going on at Spark. Next. And finally, if you haven't joined Spark yet, we hope that you'll do so, and also that you'll spread the word about Spark to others that you know who may be eligible. So that's it. I think we'll turn it over to Vincent for questions.
Okay, thank you so much. We did get a number of questions, but you can send more in if you still have any. Okay, let's so this
No, go ahead.
when we talk about genetic findings, does that mean that's when we talk about genetic findings, basically, can you describe the gene list and like how we confirm that it's a gene-related autism?
Okay. So the way that we're analyzing the data is that we have a list of genes and CNV loci. So CNV refers to copy number variant, and that just means a section of the genome that's either deleted or duplicated. We have a list of these areas of the genome and genes that are already known to increase autism risk. Some of them are really, they vary in, okay, sorry. So we have this list of autism genes that we already know about, and we're constantly updating that list. So with our own data and with data that's being published, we're able to add, several genes every couple of months. But we're constantly analyzing our data for variants that we think are damaging or disrupting that particular gene, and in those genes or CNVs, if that makes any sense. So we're looking only at a targeted region of the genome, but we're also, constantly evaluating the data and looking for new findings. So there is a lot of confidence. We're only returning things that a clinical lab would judge to be as, they call it pathogenic, and that just means disease-causing. So we aren't returning anything that's a variant of unknown significance.
Yeah, I think that helps to answer the question. If someone doesn't receive genetic results back, does that mean that that this person does not have genetic markers for ASD?
No, it doesn't mean that. Unfortunately, we can never really give a true negative result to families because we don't know everything that leads to autism. 80% of autism is thought, 50 to 80% of autism is thought to be genetic, but we're only able to identify something in 10 to 15% of families. So over time, if you submitted your data to Spark, we're constantly reanalyzing it. So if you sent us your data 2 years ago, in 5 years, we're going to reanalyze it, or every year we're reanalyzing it. So if you don't hear from us, it does not mean that your family member does not have a genetic result.
And then can you talk a little bit about the kind of analysis that we do?
Yes. Right. So we do either whole exome or whole genome, depending on the sample, but for the most part, it's whole exome. And we do a genome-wide genotyping array, and that allows us to look at something called copy number variants. So there are many different regions of the genome that are known to be deleted or duplicated in people with autism, and we're able to look at those too.
So I think someone here might have participated in another study by the Simons Foundation.
Mm-hmm.
Can you just describe some of the cohorts that we found?
Yeah, so one of the big things that Safari has done is establish these really large research cohorts. We have 2 other large cohorts and several others, but the 2 other main ones that are being referred to are one is called the Simon Simplex Collection. That's a collection of about 3,000 families that were started, that was 10 years ago, I guess. That's when that started. And if you've participated in that, that's great if you want to participate in Spark. And we ask families to just say if they've been a part of that, and that's really helpful. And we also have something called VIP, Variants in Individuals project, and that's a group of about 2,000 families who have a known genetic diagnosis.
Someone had a question about difficulty collecting saliva. Can you just explain the assisted process?
Yes. So with our spit kits that that you get in the mail, these require 1 milliliter of saliva. There is a sponge that can be used to passively collect saliva from an individual. It does take some time, and the amount of saliva that's in there that gets into the sponge does vary by person. Some people have more saliva than others. It's hard to control that. The clinical sites do have an alternative way of collecting saliva that does not require 1 milliliter of spit. It just requires several scrapes of the cheek. This is not a buccal swab. It looks like a buccal swab, but it's a new way of collecting basically saliva. It doesn't need 1 ml of liquid. You can only use those if you go to a clinical site right now. This is a very new kit, and so only the clinical sites are able to do this right now. We do hope to be able to send those to families long term, but that process will take some time to set up.
Kiely, maybe you can answer this one. Someone was asking if there are any studies being conducted on siblings without autism and maybe the likelihood that the sibling might have ASD, and if Spark might ever do studies like that.
Right. So just thinking about the research match studies that we have in the pipeline, we actually have two that are focused on recruiting baby siblings of the individuals with autism. So we know that if you have a child with autism, then you're more likely to have a second child with autism. And so researchers want to better understand sort of what is happening and what those risks are. And also focus on how can we identify autism as early as possible in younger siblings, and then get them into early intervention programs. We also have another study that we're working on right now. It's not, we haven't sent out invitations yet, but it's focused on adolescents with autism as well as those without autism to see, so there's this idea that individuals with autism, especially females, can sometimes hide their autism or mask it when they're out in social environment. And so the study that the researcher wants to do is looking at typically developing children, which includes SIBs without autism, as well as individual with autism.
Might only be able to do about two more questions. So someone was asking, even if we don't find something right now, say 10 years later and more data becomes available, would we be able to return a result to someone?
Yes.
And then a follow-up to that was, is our analysis clinically validated in some way?
Yes. So all of the samples that are initially received by a CLIA-certified laboratory, as long as there's DNA left, we use that sample to confirm, and that goes through their clinical lab using all of their certified processes, and they are the ones that issue the clinical report that goes back to the family.
Okay. And then this is going to be the last one, but I will definitely take these questions down. There were some really good ones that we'd love to share the information back with you all. We can do that on our website. So the last question is if you could just describe de novo again.
Okay, so de novo is a change that, if you think about yourself, a change would be occurring in your sperm or your eggs. And that sperm or egg that produces the child, that's where the genetic change is. So it's only in your egg or sperm, and therefore, when it creates the child also has the change. So it's not something that you, in your body, the rest of your body has. So you wouldn't have the genetic change. It's just in your egg or sperm.
Okay. All right, so that's all the time